Welcome to lecture 12 in DIY transcriptomics. This really marks the last phase of the course where we will spend the remaining 2 lectures talking about how to document the kinds of analyses we've done in a way that makes it really easy for others, including our future self, to look back on an analysis, understand what was done so as transparent as possible, and actually reproduce that. So transparency and reproducibility are 2 really critical parts of any analysis workflow. And so this is a great time to talk about those because you've done a fair amount of working in R, and it's easy to end up with a project that's quite disorganized. So we're going to try to pick up the breadcrumbs, right, of Hansel and Gretel here, to follow a trail during our analysis and to leave that trail for anybody else who may want to run that analysis as well. And that's going to involve a tool called R Markdown, and we'll be talking about that in detail in this class. So the objectives for today are to discuss the basic building blocks for assembling a figure, to understand the basics of R Markdown and dynamic documents. We'll then learn how to construct our own R Markdown document using those essential code chunks that have been at the end of each of the stepwise scripts that we've used in the course thus far. And then I'll show you the flexibility of this Markdown approach in the way that it allows us to create documents that have a very different look and feel, but really have the same fundamental concepts at play. I've provided the code for doing this, including an R Markdown template that has no code in it that you could use to fill or populate with the essential code chunks from the course. I've updated all the step scripts based on some minor tweaks that we needed to make in the essentials code chunks to make this go as smoothly as possible. So if you're going to try to assemble this R Markdown document on your own today or at any point moving forward, you'll probably want to just grab new versions of each of the step scripts to build this document so that it works as smoothly as possible. I've also, however, provided for you the full R Markdown document with all the essential code chunks sort of already filled in. And then lastly, and we'll talk about this at the end of today's lecture, I've provided another R Markdown document that generates a different kind of research output, if you will, called a flexible dashboard. I've linked to one blog post on knitr. knitr is an R package that we'll use today that pulls together code and outputs into a single document. Carl Broman's not the author of knitr, but he writes a nice article about knitr and its functionality. And there's lots of content from the author of knitr online as well that you should read. And that software developer is also now a software developer for RStudio. All right, so really, I think what we've done in this course thus far has left all these sort of digital breadcrumbs along this somewhat linear path that we've tried to walk through how to approach analyzing RNA-seq data from, we have our raw data, we have some various processed forms of that data, we have multiple analysis scripts, and that's just with this relatively straightforward data set. You could imagine if this data set was more complex, you could have even more sort of scripts and approaches. We have graphs, and we have tables, and we have many other research outputs from this. And some of those for this course data set that we've generated along the way are shown here. They include, different kinds of outputs from a multivariate exploratory analysis of the data, including dendrograms and PCA plots. Lots of different outputs from differential gene or differential transcript analysis, including tables that we get from GT, volcano plots for one or more pairwise comparisons. If you felt so inclined, you could have used the isoform switch analyzer package to look at differential isoform usage between 2 conditions. Again, not a great demonstration with the data set we're working with in class, but still something that you can have lots of figures from. We have heat maps, and from those heat maps, we capture modules of coordinately expressed genes. And then in the last course, in last lecture, we went through lots of different functional enrichment analyses, which generate many other kinds of graphical outputs very different from these, right? So we had Manhattan plots of different enriched terms from various gene ontology categories. We have enrichment plots that show the enrichment score for any given signature across our whole data set. And those are from gene set enrichment analysis. We could obviously take any of the results from a Go enrichment or a GSEA and show a table of those as well. And we can summarize those kinds of tabular results in bubble plots and other kinds of visuals. So these are really building blocks for assembling a figure. And you got a sense for this in the last Hackdash challenge, where you had to build your own figure from a data set you had never seen before. And many of you drew on these, and pulled from these different elements of the scripts, including PCA plots, volcano plots, heat maps, and more. So that's, I think, one key thing to take away from this lecture is that you now have the building blocks for assembling a figure using RNA-seq data. And I want to emphasize that this is not just for RNA-seq. Actually, none of the graphics I'm showing you here are in any way inherently restricted to RNA-seq data sets. PCA plots can be generated from numeric data of any kind. Same thing with dendrograms. Any sort of feature selection process can generate volcano plots and heat maps. Modules are also really quite generic, and enrichment analyses can be done with many different kinds of data sets. So there's absolutely no reason why these building blocks that you've learned about in this class couldn't also be the building blocks for your single-cell seq experiments or your microbiome experiments. In fact, along those lines, there's been a recent paper on bioRxiv that really addresses exactly this point, which is many of the methods we use in class are generalizable. All right, even though they were developed for microarrays or early on and then adapted for RNA-seq, or maybe they were developed for RNA-seq exclusively, there is quite a bit of evidence that many of those methods are just as useful for other kinds of data sets. And so I won't go, you can take a look at this paper, I won't go over it in great detail, but just to highlight, they take a number of different kinds of data sets, 16S microbiome data, shotgun metagenomic data from a variety of different locations and projects spanning different body sites. And they use a number of statistical metrics to assess how well methods like DE-seq or EDGAR or Lima, how well those methods perform with data that's inherently more sparse, like single-cell seq data or shotgun metagenomic data, et cetera, or 16S data. And so they formally test, and I think there's not many examples of this kind of sort of formal evaluation and comparison of these different RNA-seq methods for other kinds of data, certainly not to the extent here. And I'll just, cut to the chase here. One of the lines from the paper is our analyses suggest that DE-seq2 and Lima-VUM show the best performance for, application to metagenomic or microbiome data. Again, highlighting that there is nothing specific or inherently limited about the methods we've used for RNA-seq. They can be used for other things, which mean these building blocks can be useful for you in lots of ways. All right, so that kind of leads to this question of how do you make, if those are the building blocks, right? How do you build something, a figure that's informative and visually compelling? I feel like this is a topic that could have a whole course developed around it. How do we make figures from genomics data? I think it's a fascinating topic, and it's one I sort of think of anytime we're talking about a large data set and how it's represented in a journal club paper, for example. What are the elements of the figure that I like? What are the elements that I think are confusing? It's not something we're formally trained in science, is sort of the graphic design aspect. And I'm not just talking about aesthetics, but how we create graphics that are clean, informative, and visually appealing, right? So I'll give you a few of the tips that I've sort of come across in my experience. And I will say right away, I'm no expert in this, but it's an area I think we can all improve in. So the first thing I would say is great figures for a paper do not necessarily equal great figures for talks. And so that means you have to often have different versions or flavors or forms of your data analyses or data visuals for different settings, right? And that's another reason I think R or any command line language is really great for this, because it allows you to iteratively refine and modify figures for different settings or audiences. Interactive plots are a really valuable tool for communication. We've used Plotly in the course and the data table or DT package to make interactive figures and tables respectively. And I hope you've come away with a sense that those are really cool for being able to just kind of interact with your data in real time and for have others interact with that data without ever having to have run the code. Of course, those interactives are not yet the kind of thing we can publish in a paper, but there may be a time when that's the case and you're already in a position to be able to produce those. I think when it comes to genomics data, making good figures is as much about what you leave out as what you put into the figure. I think one of the most common mistakes when trying to make a figure with say an RNA-seq dataset or a ChIP-seq dataset or a microbiome dataset is to include all findings. And that's not always a good idea, right? We know that figures are supposed to have a clear message. And so if you have multiple different messages evident in a figure that you're trying to emphasize, the figure can become overwhelming and confusing, and your reader may miss actually the main point of the figure. So think about not just what you want to put in, but what's better left out, maybe included as a supplementary figure, maybe not included at all because it really requires a totally different kind of analysis to really flesh out. I think a good figure distills your data. I think it starts, many good figures start broad and they quickly funnel down to a salient finding that entices the reader. So a good figure distills your data. I also think a good rule of thumb is to show your reader something familiar or expected before showing them something new and unexpected. I think this helps build trust and confidence in your figure and your analysis. You can think of this as the sort of positive control element in a genomics figure. If you're doing an RNA-seq on a cell type or tissue type, chances are other people have some knowledge of that tissue type or cell type and have some expectations of what should be found. So lead with that. Show them that indeed your analysis reveals things that are long-held paradigms or concepts in the field. That will help build trust. Then show them something new, different, perhaps surprising. If you lead with the most surprising result first, sometimes it causes the reader to doubt the data itself and to doubt your analysis. And then I hope you've gained some understanding throughout the course that some visuals, although maybe standard visuals that many people use, they have inherently misleading aspects about them. Two examples we used in the class are heat maps because of the colors. If chosen incorrectly, they can cause problems for those with color blindness, but also because the colors in most heat maps, if people have scaled the rows, do not reflect the actual data, they reflect z-scores, and we need to understand how that changes the interpretation of the heat map. Venn diagrams, also misleading because they suggest that genes fall into neat bins where they're either differentially expressed in one setting, differentially expressed in the other, or both or neither. And in reality, we know that genes can approach a threshold for being called differentially expressed, but not meet that threshold. They then end up in a bin, right? Even though they actually were really close to being differentially expressed. And so taking a Venn diagram and making a heat map from the genes in that Venn diagram sometimes leads to a disconnect. And so it's just important to be aware of that. And then finally, take advantage of these building blocks to create at least a first draft of your figure. You know I'm no fan of Venn diagrams probably by this point, and I always love this paper. It sort of turns out to be a great mathematical puzzle of how to make a symmetric Venn diagram that includes 11 Venns. I love the title, the first simple symmetric 11 Venn diagram. Just to highlight with any Venn diagram, the number of zones, the number of sort of areas that you see shaded here is 2 to the n, where n is the number of Venn circles. So for a data set that has 4 Venns, 2 to the n is 16, right? So 16 different regions that you might have to discuss in your paper. The malaria data set that we were dealing with before had 5 different groups, so a huge number of possible comparisons. So that's led some people to develop, this idea of other ways of viewing intersections between groups in a way that's more informative than Venn diagrams. This is something that's been termed an upset plot. You may see it. It is a better way to represent Venns, but it comes with its own complications in that it's incredibly dense and takes a bit of time if you've never seen them before to kind of walk through. But basically, you can see for any, this is 5 different groups, so this would have been a 5 circle Venn diagram. For any groups, you can see the number of genes that intersect for all possible intersections. So for example, this is all genes differentially expressed in both P10 and RB1, and that's only 5 genes. Whereas this are genes that are EGFR regulated and P10 regulated, and that's 16. And they're rank ordered by the intersections that have the most genes in them. And then of course there's lots of portions that are EGFR only. In this case, whatever number of genes here, or TP5, TP53 only, or P10 only. And those are indicated with just single dots. So you get the idea. Just think carefully about how you use Venns. And please don't use Venns to represent functional enrichment results. It's common, but it's just wrong. So in this case, what the concern or the issue we have is by representing this data as a pie chart, we're implying that genes annotated with the Go term immune response, which fall in this yellow slice, are only annotated with the term immune response. So that's what's implied with a pie, right? That you belong in one slice. But we know that genes are annotated with many Go terms, and there's a lot of redundancy. So I can guarantee that the genes found in this pie slice are probably also in this red one, right? They're annotated with the Go term immune response and defense response, and probably response to biotic stimulus and response to stimulus. So there's a lot of redundancy that is just not appropriate to represent as a pie chart. So I'll show a few figures. We don't need to spend a lot of time on this, but just to highlight that you don't need to explain all of your data in a figure. So as an example, we had worked on a paper that shows each row here is a different transcriptional state due to different kinds of parasite infections. And we can see a set of genes here that I've indicated because I want to talk about them that are highly induced by one kind of parasite and which involve a lot of canonical antiviral genes. This is my sort of positive control, right? Here's a bunch of antiviral genes. You recognize these. And so no surprise when we do Go enrichment analysis, response to virus is one of the key things that comes up, indicating that this parasite has an immune response that's generated that highly resembles that of a viral infection. That's not to say, and this is why I say you don't need to explain all of your data, there are lots of other interesting modules in this data. Here's one, here's another, that seem to hint at unique biology and indeed are able to reveal some unique biology, but they're not the topic of this figure, so they're not discussed. And in a way, this can almost bait reviewers a bit in that if a reviewer really wants to know what this module is that's induced by this other parasite and not by the one we've highlighted, that's a great way to respond to a reviewer's comment, do an analysis, either show it in the rebuttal or include it as a supplementary figure. I think it makes sense to kind of march through your figure with the goal of distilling your data. And remember, I mentioned this in the previous slide. The idea that we start big, all differentially expressed genes. We focus on a few modules, which I've shown here. We carry out Go enrichment for those modules. And then for the key modules, we show the top genes. And I think this is something that reviewers often look for is gene. They attach meaning to gene symbols. So it often helps if you can get beyond large scale analyses to actual genes, even if it's just a top 5, 6, top 10 gene list. And if you have genes that, again, serve as sort of positive controls to ground your analysis, it's great to highlight those. In this case, genes that are known to be involved in CNS function previously, and these are some perhaps new ones, that can really help the reader as well. And of course, now you can combine your differential gene expression and module identification with functional enrichment analysis, but also take your entire data set and do Go enrichment. And then from those, GSEA plots or from the modules themselves, you can show top genes, right? You could use dplyr to rank these by log fold change and produce an a priori heat map just from those genes, right? These are all things that you can do now. But there's other ways to show things, and not all types of figures provide the same value. This is a figure from a science paper where, again, you recognize a volcano plot and the idea of selecting upregulated and downregulated genes with respect to one pairwise comparison, in this case, gestational colonization and control. But then the functional enrichment they carry out is done using something called String, which is an online database resource. And they show the genes and these sort of networks they form for, Go terms. So, this helps them reduce these upregulated genes to a set of enriched Go terms. Although this is useful, what's kind of lost in this figure is I don't know what this gene is. All right, I don't know what the most upregulated genes are. These networks don't really tell me anything here. They're not telling me maybe what the biggest changers in a data set are. So they're sort of visually striking, but they may not be as informative as sometimes a table. All right, I'll close this part of the video just by highlighting that many of the students from this class in the past have used these building blocks to create their own figures from their own data during the class or shortly after that have then gone into a number of different papers. This is Brian Gaudet, who was in the class and really took off and developed a lot of skills beyond what we did in the course to generate his own figures. But you see a lot of the elements that you're familiar with, heat maps, GSEA plots, volcano plots with certain genes highlighted. Corbett Berry, who was a former TA from the course, did the same thing. And then former students in the class, including Naomi Phillip from Igor Brodsky lab, now a postdoc in Ruslan Medsetov's lab. Again, you recognize many of these building blocks and on and on. So we have a lot of students who have done this, and I encourage you to take this time to start to construct your own figures. Okay, so we now understand this idea of building blocks for assembling a figure, and now in the next part of the lecture, I want to talk about how we actually start to wrap all of this stuff together in a reproducible document.